We're going to continue on with the nonspecific immune response and take a look at the phagocytes. Remember, there are 4 different phagocytic white blood cells. So they include the neutrophils, the monocytes, macrophages, and dendritic cells. So that's the neutrophils, monocytes, macrophages, and dendritic cells. And all of these 4 cell types are part of the nonspecific immune response. So they will go after and eat anything. They don't care what it is, and they're not going to be picky. So the 4 phases of phagocytosis are chemotaxis, where the phagocyte is going to follow the chemical scent trail and figure out where the microorganisms are located. Adherence just means that then the phagocyte has to grab onto the microorganism, and then ingestion, they'll just reach out their cell membrane and surround the microorganism, and then bring them inside and digest them. The first phase of phagocytosis is chemotaxis, and this is where the white blood cell is going to follow the chemical scent trail and figure out where the microorganism is. So you can actually see chemotaxis going on in this video here, where we've got this macrophage that is following around this bacterial cell and then finally catches up to it and ingests it. So once the phagocyte finds where the microorganism is, then it needs to attach to it and bring it inside. So there's 2 ways for the phagocyte to grab onto the microorganism. So it can use the pathogen recognition receptors, which are the toll-like receptors. And remember, those recognize the PAMPs, and that's the pathogen-associated molecular patterns. So those are things found on microorganisms that are never found on our cells. So things like peptidoglycan cell wall, lipopolysaccharide in the outer membrane of the gram-negative bacteria. So if the phagocyte finds any of those PAMPs, it knows that it doesn't belong in the body and will go ahead and phagocytize it. And then the other way to grab on is to use opsonins. So let's just take a look at a picture of these. All right, so here's the 2 ways to grab on to the microorganism. So we could use the toll-like receptor, which recognizes those PAMPs, the pathogen-associated molecular pattern that are only found in the microorganism. So if the phagocyte finds one of those PAMPs, it knows that it needs to eat that microorganism. The other way to grab onto the microorganism is to coat it with the opsonins. So remember, opsonins could be anything like antibodies, which I have pictured here. They could be complement proteins or acute phase proteins. And so they just make it easier for the phagocyte to grab onto the microorganism and then take them in. And then the third phase of phagocytosis is ingestion. So once the phagocyte is grabbed onto the microorganism, it will just reach out its cell membrane and then surround the microorganism and then just bring it inside. And then digestion, we're going to fuse the lysosomes with the phagosome. And remember, the lysosome has digestive enzymes inside of it, so then that will break down whatever was taken inside. The lysosomal enzymes also create some reactive oxygen species as well, which, remember, those are pretty toxic to things. Okay, and then once they're done ingesting and digesting whatever it was that they took in, the neutrophils will actually expel any extra fragments by exocytosis, but the macrophages and dendritic cells are going to do something really different with what they've eaten. So they're going to take a piece of what they've eaten and they're going to present that antigen to the T lymphocytes. And we want to see if any of the T lymphocytes recognize what it took in, and then if we find a match, then we can activate the T lymphocytes. So here's just a picture of the antigen-presenting cells. So remember that the macrophages and dendritic cells, both of those are antigen-presenting cells. And then the other one that we're going to talk about in the next chapter that's also an antigen-presenting cell are the B lymphocytes. So macrophages, dendritic cells, and B lymphocytes, those are all antigen-presenting cells. So here we have, for example, a dendritic cell that is phagocytized some bacteria. It's ingested and digested it with the lysosomes, and then it's just going to take a fragment of that bacterial cell and then stick it out on its cell surface and then go around and present that to different T lymphocytes to see which T lymphocyte recognizes that bacterial cell. And then we can activate the T lymphocytes. There are some microorganisms that can actually resist being eaten. Remember we talked about biofilms with that slimy layer that gets formed over the bacteria. The phagocytes can't get through that to get to the bacteria. And then remember we talked about bacteria that had a capsule outside of their cell wall. So remember the capsules are very slippery, and so it's hard for the phagocyte to grab onto them. And then some of the bacteria are actually very sneaky in that they're able to avoid being digested once they get taken inside. And so they just end up living inside the phagocyte. So over here on the right, we've got a picture of some macrophages. And stained in orange are the bacteria Mycobacterium tuberculosis, so the bacteria that cause TB. They do get phagocytized by the alveolar macrophages inside the lungs, but those macrophages are not able to digest those bacteria, and then the TB bacteria just end up living inside the macrophages. And so it's really hard to get rid of those bacteria. So that's pretty sneaky. Okay, and then this is just a picture of the cell envelope for Streptococcus pyogenes, which is the bacteria that causes strep throat, among some other things. And Strep pyogenes does have a capsule that's kind of in purple here, and that makes them slippery. And then they also have this M protein that sticks out, and that actually prevents the complement proteins from sticking to the Strep pyogenes bacteria. Because remember, the complement proteins are opsonins, and they normally would coat the bacteria and make it easier for the phagocytes to grab onto them. So Strep pyogenes is pretty hard to phagocytize with the capsule and the M protein. I won't ask you about the M proteins, but we'll talk about those a little bit later. Those will come up again.